Hey guys, so here's our next set of notes. Coming up, there we go. Had to get everything set up the way I needed it to share the screen. So now I have it ready. So we'll get that shared and put it in present mode. So we're continuing looking at some of the subcellular components and compartmentalization. This time we're focusing on the cytoskeleton and compartmentalization. Okay, so cytoskeleton is basically, it's just a network of fibers. Just like we have bones, our cells have a structure to help give them support. So they're just not like a flattened sac, but they're not bone. They're made of a cytoskeleton. So they're all about giving structural support, especially in animal cells. Plant cells, because they have a cell wall, tend to have their own built-in support with that. But animal cells don't. So that's why the cytoskeleton is especially important with animal cells. It provides mechanical support. It actually helps anchor the organelles so they're not just randomly floating around within the cell. And it also allows for movement of the vesicles and organelles, or even the whole cell. We can use these kind of cytoskeleton structures almost as like tow ropes or things like that to move things around. Movement occurs when the cytoskeleton interacts with motor proteins. Those motor proteins are actually what helps it move. Think about, you have a boat that can move around, but until you put the motor on it, you can't really control that movement. So the specific fibers that we use when we talk about the cytoskeleton, we have microtubules, microfilaments, and intermediate filaments. Okay, so we're going to start with the microtubules. They're hollow rod-like structures made of a protein called tubulin. So think of them kind of as like a little straw. They grow from the centrosome, also sometimes referred to as the centrioles. And this will actually assist in microtubule assembly. So this is going to help build more microtubules. So they really are all about structural support. They're almost kind of like tracks that you can move organelles and things along, almost like train tracks that the train travels specifically along. These are going to be similar in the way they behave when they're interacting with those motor proteins. They will also, and this is a big one, assist in the separation of chromosomes during cell division. So when we get to talking about mitosis, we're going to talk about the spindle fibers, and specifically spindle fibers are microtubules. They also play a role in terms of cell motility, so they're a part of cilia and flagella in particular. Okay, the next one is our microfilaments. So these are thin solid rods. So these would be more like a little bamboo skewer that you might use to put food or something on, because they're solid all the way through, whereas the microtubules were more like little straws. So these are made of a specific protein called actin. It's one of the key materials that allows your muscles to contract. So these are all about maintaining cell shape. They can deal with, when it says bare tension, what it means is they can handle a lot of pressure or push or pull a lot of tension on them. So they're going to assist with muscles contracting and also with cell motility. So they'll play a role as well in terms of flagella and cilia, things like that. Actin typically also works with another protein called myosin to form that contraction. When we are dividing the animal cell, the contractile ring that forms the cleavage furrow is actually made of microfilaments. And then we have our intermediate filament. So these are fibrous proteins with lots of different subunits that make it up. We're going to find these as permanent structures within the cell. So we're not going to assemble and break them down like the microtubules and microfilaments will be. These basically, once they're built, they're going to stay there until they're just no longer needed for any reason. So these are really about maintaining that cell shape so it's not just collapsing. And this is Important in terms of anchoring the nucleus and other organelles within the cell. And it also forms the nuclear lamina, which is kind of the lining for that nuclear envelope. Okay, so we have a free response. You're working in a research lab and your goal is to determine the effects that alcohol has on the liver. You design an experiment where you expose samples of liver tissues into varying concentrations of alcohol as follows. And so here's the chart. After exposure to the alcohol, you analyze the liver samples. In all samples, you find that the alcohol has been broken down or detoxified. However, in sample 3, you find damage to organelles has occurred due to the high concentration of alcohol. So A, identify one organelle in the liver that could be responsible for the detoxification of the alcohol in all 3 samples. B, in sample 3, identify the organelle that would most likely be responsible for removing the damaged parts of organelles. And C, describe the steps it would take to do so. And then we want to focus on the idea of compartmentalization. And so when we talk about compartmentalization, what we're talking about is it's almost like breaking one single building up into separate rooms or areas. So by breaking it into different organelles, we can actually have a lot more metabolic reactions occurring and we can control where they are occurring. So it's not just throw everything in there and see what happens. It also increases surface area because each compartment and space provides more surface area for reactions. It keeps reactions from one place interfering with reactions that might be occurring next to them. Because if we can separate them into their own little areas, even if they're in the same location, if we got them separated into their own kind of areas, they won't cross and mess each other up. And then, of course, we've been talking off and on about the endosymbiont theory. That's that one that kind of is trying to explain the mitochondria and chloroplast, where their own freestanding prokaryotes. So here's the official theory. An early eukaryotic cell engulfed a prokaryotic cell. So that prokaryotic cell then became an endosymbiont, a cell that lives in another cell, and they became a single functional organism. The double membrane, because we have our natural membrane, and then when it was engulfed, we put another membrane around it, that would lead to the double membrane. It has its own ribosomes, which we know all cells have. And it has circular DNA, which is more like what we find in prokaryotes rather than in eukaryotes. And we know that they can actually function on their own, even if they don't get anything from the cell, they can still do what they need to do. Other practice free response. In humans, pancreatic cells synthesize the protein insulin in the endoplasmic reticulum. Patients diagnosed with type 1 diabetes have immune systems that mistakenly attack their pancreatic cells. Therefore, these patients cannot produce the hormone insulin and have to take medications to introduce insulin into their bodies. Describe how insulin would be produced in a normal functioning, non-type 1 diabetic pancreatic cell. Okay, so hopefully that's going to help you out with those a little bit. And I will see you in the next video. Bye.